Hello and welcome to today's webinar entitled Just Physiology, Human Physiology Simulation Software Designed for Students. My name is Dr. Patricia Halpin, and I will be serving as your moderator today. I'm the chair of the American Physiological Society's Teaching of Physiology section and associate professor at the University of New Hampshire. In today's session, Drs. Robert Hester and David Julian will demonstrate how physiology educators may use the JustPhysiology platform to allow undergraduate, graduate, and medical students explore human physiology using in-class and self-paced activities. I would like to thank our sponsor, HC Simulation, for working with APS to spotlight innovative educational techniques and tools. Now for a few housekeeping notes. Out of respect for our speakers, we ask that you refrain from photo or video captures of the presentation in whole or in part. This session will be recorded and available for future viewing through a link on the registration website. Recording or taking photographs of another person's presentation without their explicit permission is prohibited. At this time, all attendees except for the speakers are in listen-only mode with the video off. As moderator, I will be happy to raise questions that you type into the Q&A function located at the bottom of your screen. You may enter questions at any time during the presentation. The question period will be answered at the end. This session will end at approximately 2:15 p.m. Eastern Time. It is now my pleasure to introduce our speaker, Dr. Robert Hester. Dr. Hester is the Billy S. Guyton Distinguished Professor in the Department of Physiology at the University of Mississippi Medical Center and Director of the Centers for Computational Medicine. Take it away, Dr. Hester.
Thank you very much. Dr. Julian, would you advance this slide? So I also have a startup company called HC Simulation. We'll be giving this presentation with Dr. David Julian, who's been a collaborator for several years with that. Next. Disclosure other than me having an excellent company is Dr. Julian has equity in JustPhysiology LLC, which provides this software. Go ahead.
Okay, folks, so we'd like to start off with some just kind of understanding who the participants are. And Mr. White has brought these up. If you would please enter information. This is all anonymous, of course.
And while you're answering that, I'll give a little background on Dr. Hester. Dr. Julian. Thank you, Dr. Hester. Dr. Julian is an associate professor in the Department of Biology at the University of Florida, where he teaches cellular and systems physiology in the biomedical engineering program.
That is true.
Mm-hmm.
Okay, great. So the vast majority of people here consider themselves physiologists, and we have some special guests who are not themselves physiologists, but are here presumably because they teach physiology classes. Thank you all for answering that. All right, let's go to the next question. Want to try to understand the types of students that you teach, and there may be more than one of these. So if you would please find which is the most appropriate for the students that you teach in the physiology course is relevant to this. Okay, great. All right, so almost equally among undergraduate, master's, and professional school, and some community college, and others. I'd be particularly interested in hearing what the others are. If anybody wants to send me that in an email, that would be cool. All right, thanks a lot. Okay, now the last thing we want to know about is the format of your teaching. And what we're really interested in is not so much the lecture part or your lessons that you present, but the parts where you might have activities where the students could work with, where they might do laboratory experiments or dry labs. Or you may have no lab, in which case it might just be all remote or virtual. Okay, great. All right, so about a quarter have no lab, a quarter have remote or virtual labs. I'm sure those became a lot more common over the last couple of years. About a quarter in-person labs, and then obviously there's more than 100% here. Some people do multiple types of labs, some with students as subjects, and then still 30% with some animal subjects. All right, great. So with that in mind, what we want to talk about is how Just Physiology might be used in these contexts. And I'll give it back to Dr. Hester.
So thank you very much again for attending. This project, Just Physiology, is based on the original Guyton-Coleman model that was developed in 1972. So basically, we have 50 years worth of advancement of this. And next slide, please. It is called Humod. It is a deterministic physiological model. It's full of mathematical equations. It has about 8,000 variables and 2,000 parameters. There are the organ systems: left, right heart, lungs, left, right kidney, liver, skin, skeletal, respiratory muscles, full endocrine system, a lot of focus on cardiovascular system, renin-angiotensin-aldose system, ADH, ANP, and insulin glucagon. We'll give you a little bit of a presentation with insulin glucagon in just a little bit. It has a pretty active nervous system. Okay. So originally this was done as a program called QCP. You can actually go to HumMod, HUMMod.org, and download this particular version. The challenge with that is we cannot modify it. It's compiled code, and this is actually where David Julian came in probably 6 or 7 years ago, maybe a little bit more. Say it's really nice, but we need to do something different. So that's when we moved to Just Physiology. Next slide. And so basically, this is the version of Humod that we use routinely. Is actually on servers, and you can use a browser base to run simulations that we have created. Okay, David, your turn.
Okay, thanks. And so what I'm going to do is kind of model a teaching activity that I might do in my class. And I actually do this in my course on the first day. And I start off by, of course, talking about the fact that a lot of the course is going to be about homeostatic regulation. I show this slide just showing the feedback loop that's involved in regulating blood pressure. My students are biomedical engineering students, so they love hearing about feedback loops. But of course, we talk about this in any physiology class. And what I want the students thinking about then is, okay, what is their current knowledge about how this works? What is their understanding of the material, and how can we pique their interest in thinking about regulation in a more sophisticated way? So I asked them to think about exercise and how the increase in power output that is necessary for exercise, or that is a component of exercise, affects all of these physiological parameters: metabolic rate, arterial PCO2, PO2, sweating, core temperature, blood glucose, cardiac output, and total ventilation. Now, many of these things are not familiar to the students yet. They may or may not know much about arterial blood gases, cardiac output, and total ventilation, but I'll talk a little bit about what these are. You know, this is the volume coming out of the heart. This is how much you're breathing. Okay, so in my class, I have the students work in small groups. And so I might ask them this question. Now, when I would do it in my class, I would actually ask them, which of these parameters, if you exercise, would go up, which would stay the same, and which would go down? But because we're on Zoom, we have a limitation. So Mr. White is going to put up a slide that's just going to ask you, which of these parameters do you think will go down when a person begins to exercise at a moderate level? Now, you can answer as you think your students would answer, but I think it's reasonable just to answer what you think. Which of these, if any, or all of them, will go down when you exercise? So we'll take a minute or two. It's going to a bit to think about here. Okay, I think we might have enough responses, or let's take a look. Okay, so this actually pretty closely mirrors what I see in my class. And so the majority of people felt that blood glucose concentration goes down with moderate exercise, that arterial PO2 goes down, and some said that 15% said arterial PCO2 goes down. And as I say, this is pretty much what I see with my students. And this gives, of course, an opportunity to think about, okay, why did you think these things? What do you expect to happen? All right, so then what I like to do then is to say, okay, now let's do an activity. And we'd like you all to do this activity. So go to the site, JustPhysiology.com, and you'll see the opportunity to, at the top, try a free case study. And you don't need to be a registered user. You don't need to log in. If you do have access, you can log in, but you don't need to. And you'll click on the. This will open up an intro to exercise case study, and you can click on integrative responses. And then, so I think everybody has seen that. Now what I'm going to do is I'm going to share my screen, a different screen, so we can kind of go through this together for those of you who might not be in front of a computer. So let me. Okay, you should be seeing something about like this. And what we're going to do is we're going to click on the second module here, which is integrative responses. And that pulls up the simulation. And here, which we typically do, we try to in most of our activities, if we list all of the variables that students could monitor. Now, not all of these are displayed. We've picked a subset. But we've got, there's always a y-axis here, an x-axis. I have zoomed out. This is written so that it works well on a tablet, but I usually use it at around 70% or 80% zoom. And you can do that in your browser. And your browser always remembers the zoom level, so you can always come back and see that. So I'm gonna actually zoom out one more level so we can see more figures. And I'm gonna run a 1 minute simulation. And I'm going to use it at 0 power for the bike. And I'm just going to run this. And so what we're doing now is we're establishing baseline levels. So we can see the bike power is at 0. We're looking at metabolic rate, blood pressure, cardiac output, total ventilation, arterial PO2, arterial PCO2, sympathetic nervous system activity. This is a relative activity value, doesn't necessarily correlate to action potentials per second. The blood glucose, core temperature, and sweating rate. Okay, so now I ask the students to set the bike power to 150 watts, which is a moderate level of exercise. And I'm going to run that for 10 minutes. And we're now going to test our predictions. And the simulation will run. Everybody who does this is running it on a server, so everybody has their own instance. We're seeing this is the bike power setting. We're seeing the metabolic rate going up, slight drop in blood pressure, which then comes back up. Cardiac output rose a lot. Total ventilation rose. Now, here's one of the first things that many of you thought would go down, or many of you said your students would think would go down, would be the PO2. But in fact, it doesn't. Let's look at another one. Many of you guessed that, or said that your students would guess that the glucose level would go down. It doesn't. In fact, it goes up. In fact, the only thing that goes down is the arterial PCO2, which the majority of you thought would stay the same or would go up. And so this is an opportunity to really talk about homeostasis, because what's happening here is when the students answer that most of these things will go down, it's, of course, it's a misconception. It's because they're understanding the first half of that feedback loop, which is the demands placed on oxygen gas exchange, the demands placed on delivering glucose. But they're forgetting about the second part, which is that the body has mechanisms in place to maintain these constant or even slightly elevated. And so that sets the stage then in a lot of the rest of the class. What I'll spend time going through more simulations to try to understand how these things are regulated, why are they regulated, what are all the feedback loops involved, what are the sensors involved in monitoring these things, and many of these things we can do in Just Physiology. Okay, so hopefully you all have a chance. You can go ahead and play with that. You can do like my students do. They can do with higher power. You can actually increase the ambient temperature, and you can look at how that affects sweating or any of these other parameters. So actually, there's quite a bit you can do just with the free version here. Okay, so let me go back and continue displaying. Okay, so as we saw, the only one of these things that went down was arterial PCO2. And in fact, the assumption might be, why doesn't it go up? Well, the reason it doesn't go up, of course, is because the ventilation is so high to take care of the O2 requirements that we end up blowing off more CO2 than we produce. Okay, so how do I use JustPhysiology? I've had a number of years of trying to figure out different ways to do it. It can be used for instructor-led demonstrations, something like what I just did. And we've done this, I've done this in classrooms with hundreds of students where they don't all have computers. So I'll show it up on a screen. I'll ask the students for their predictions, and then we'll run through so they can see what the responses are. I do it in class with active learning exercises. So for example, like with a flipped classroom format where the students have clickers or some kind of classroom response system. Breakout sessions, dry lab sessions. I saw somebody in the chat pointed out that we didn't, in the options of how your lab would be, we didn't talk about small group breakouts. In fact, that's a way that I've used that. I use these in self-paced lessons where I'll give them assignments to do, and they can work on this from one class to the next, or they'll have a week to work on these. We can do this in a fully online learning format where it's all self-paced lessons. We don't have a lot of these set up, but you could build them into your classes. And as I'm sure Robert will explain, we're happy to build special modules based on interests that people have. And in fact, as we'll show you a little bit later, we can use it for authentic research, where the students actually do experiments, they design experiments, they gather data, they analyze the data, and they write that up. All right, so as an example of an activity students could do on their own or as an instructor-led demo, I'm going to hand it over to Robert. You're muted.
Sorry. David, you need to unshare. Okay, so this is actually a baseline person. This is a normal person. We're going to do an oral glucose tolerance test. We give some directions here. Basically, this person has gone 6 hours out and they have been fasting. So if you look over on the right side, we have their diet intake fixed. They have basically 0 times normal. We've gone 6 hours. What we're going to do now is give them an oral glucose test. And we'll do it for just 2 hours just to minimize time. And what you'll see is it takes just a moment, and now you see glucose starts going up. You see insulin goes up to take care of that. At the same time, plasma glucagon as a feedback mechanism goes down, and our liver glycogen is going up because we've added this particular glucose. So we can go for another 2 hours fairly quickly, and you can see what ends up happening. There's a fairly short drop off there. Our insulin's coming back down, and our glucagon is coming back up towards normal. So we've done a little bit in terms of adding liver glycogen back during this glucose tolerance test. Now, we actually have situations where we can set a value where it doesn't change. So this is actually, I'm just going to show the results. This is a scenario where I did exactly the same thing. Diet intake was fixed. We had a dietary goal of 0. We did an oral glucose tolerance test, but note here is insulin never changes. And what you saw, if I go back to the previous slide, is glucose went up to 125 here. When you fix the insulin, glucose goes up to 100, almost 175. We don't have a whole lot of falling glycogen there because we're not adding a whole lot. So this is sort of an interesting thing from a hormonal aspect. One of the things we didn't mention is you actually see 3 different little buttons right here. And when David talked about research, if you actually click on this one, it will expand it to let you see it in a little more detail and all the data points. And if you hit export, you can actually close it out into a CSV file to look at it a little bit later on. This one just allows you to move the graph if you want to place it. This one allows you to get rid of the graph. You're actually able to add graphs depending on what you want to. The list that we have up there, but we have all these covered right now with that. Okay, so that gives you some idea about the ways in which you can use the simulation showing the effect of basically fixed insulin on plasma glucose and the hyperglycemia that is observed. The next one I want to show is actually one that David has actually used before, and I actually use it for my medical students as a challenge for them. It's a little bit long in terms of introduction, but basically I'll say briefly, you're hiking with a friend when they have an injury, which causes internal bleeding. You cannot stop the bleeding, but you have a list of treatments. How long can you keep your friend alive? Discussion about this, and here are the things that you're able to do. You can give them sodium chloride. Hetastarch is actually a volume expander. It's basically sodium chloride with a colloid in it to stay inside the circulation. Epinephrine, norepinephrine, and insulin. So I ran it for a period of time. What you can see is blood pressure is starting to go down, blood volume is going down. If I run it for, say, an hour, what you'll see is these things will continue to go down. We actually use it also as an index to show that the tissues are being perfused. David decided urinary output should be above 0.2 mils a minute to show that the kidney is being perfused. And you can see right now we've got a low blood pressure, heart rate's going up, our blood volume has dropped almost a liter, and our urinary output is going down. So these are the things over here you can actually give them. We didn't mention oxygen. In fact, oxygen becomes very important when your hematocrit goes down because you can transport some amount in a dissolved state. So this is one of the things I use with my medical students. I actually use it as a competition to see who can keep the person alive the longest. But that sort of gives you some value, some idea of the breadth of the particular model and another way to particularly use it. David, I think it's back to you now. And I will stop sharing. Stop sharing. I've stopped.
I'll talk a little bit about how I use an activity like that in my class. Obviously, that's not something we do in one session. That's something students work on for weeks. Let me show you one other activity that I've did last semester. So I asked the students to create a cardiovascular regulation diagram. Once again, this is getting at this idea of what are the things that are sensed and regulated. So the students are challenged to conduct experiments to create a diagram showing autonomic nervous system regulation of the cardiovascular system. And the controls for this are they can regulate the sympathetic nervous system activity, they can regulate the parasympathetic nervous system activity, and they can regulate the exercise intensity. So I'm not going to show the simulation in real time, but I'll just talk about some of the results. And I don't tell the students how to conduct the experiment. I just give them the model and they can control any of these things to try to make the connections between all of the variables. And there are a lot of variables, and this takes them an hour or more to do, and they do this in groups. So here's an example of some of the data that they can generate. Now, in this particular case, the experiment that's shown here is where the sympathetic and parasympathetic nervous system are clamped at a constant level. And then at the 5-minute point, the sympathetic nervous system activity is increased. So we are stimulating it. And so the students then can observe which are the parameters that responded when the sympathetic nervous system activity increased. Okay, so we see an increase in heart rate, we see an increase in cardiac output, mean arterial pressure, contractility, end diastolic volume, vascular conductance goes down, so et cetera, going through some of these things. Now, the students, of course, have to eventually learn that some of these are direct effects, right? Some of these things have adrenergic receptors, and some of these are indirect effects, right? And so they have to start thinking about how these things are fit together. And the idea is to say, well, this directly regulates that, this indirectly regulates that. Another experiment they can do is the same thing, but they increase parasympathetic nervous system activity. Now here they see heart rate goes down, stroke volume goes up. Okay, that's a little bit, that causes a lot of them a challenge, and it's counterintuitive until they start thinking about all of the systems working together. Cardiac output goes down, mean arterial pressure goes down, etc. And I won't go over all of these now, but you can go through these. It's an activity that we have available. And then finally, what they do then is they will, sorry, I went backwards. They'll do exercise, but with the autonomic nervous system clamped, and then they'll bring in the autonomic nervous system regulation. So for example, we first start exercising only at a low power because the body doesn't have the ability to respond with the sympathetic nervous system. So we're going only up to 50 watts. And here's an interesting thing. Right away, we're seeing the mean arterial pressure drop. So there's no autonomic nervous system activation. We're seeing muscle blood flow go up. We're seeing metabolic rate go up. So the students who do this experiment in this way have the insight that, well, these things then are direct results of exercise. They're not dependent on autonomic nervous system regulation. And then they see that other things like heart rate doesn't come up, okay? Stroke volume does. Cardiac output does a little bit. Then they increase the sympathetic nervous system to a level that they've seen is consistent with that level of exercise before. All right, now they see the heart rate come up. Then they decrease the parasympathetic nervous system and they can see the full response that they normally see with exercise. Now, this is obviously it takes a long time. These are fairly advanced students at this point in the class. They've gone through most of the semester, but they're able to come up with fairly complex diagrams of how these systems work together, and it allows them to think about beyond just simply the feedback loops and more into cause and effect. And we find that, the mean arterial pressure is going down because we're getting an increase in muscle blood flow, which is increasing the conductance and decreasing the peripheral vascular resistance. Whether you go over it at this level with your students is up to you, but this is the level that you can go into in terms of the complex interactions in the cardiovascular regulation. Okay, so other ways that I will use this is I have the students do research reports in some semesters, and this is something that takes up about half of the semester. They do it in stages, so I give them a question that they need to use JustPhysiology to research. I'll set up a lab for them where it has all the variables they need and all the controls. Robert and I can make one for you if there's something you're particularly interested in investigating. Some things don't work in the model, but a lot of things do. So one I did was ask the students to conduct experiments to determine the optimal strategy for emergency stabilization of a patient with internal bleeding. That's the hypovolemia treatment challenge that Robert talked about. So the students will have an opportunity to spend a couple of weeks with this. I have them submit a proposal, and then they do their research report. And then I use this with a peer review format. So the students do anonymous peer review, and then they do a second round of reports. Some of the reports are really quite complex. I ask for 1 page of writing and 1 page of figures. They can download the data. They can make the figures in whatever software they find useful. Another one was to develop an experimental model for chronic hypercapnia. And this was to test the common thought that if you give somebody who has chronic COPD, if you give them supplemental oxygen, that can lead to a reduced respiratory drive and further hypercapnia. I think this is still frequently taught to clinicians or medical students. There's actually very little experimental evidence for it, and we can show in Humod that you can model this and show basically what this is false. Develop treatments for 2 patients with mild hypertension. So one patient will have essential hypertension, another patient will have hyperrenin secretion, but the students don't necessarily know that, but they're given an array of drugs that they can give. I should say that the model includes, I don't know, Robert, maybe 20 different frontline drugs. Many of the drugs that you would use in an ICU or a critical care unit, you can administer. can develop a treatment for a patient with fatigue, design an experiment to determine the effect of dietary sodium on the development of hypertension. How important is sodium? Does too little sodium cause the J point where you actually get a reflex hypertension in response to inadequate sodium? And there are a number of other things that you can do like this, and I'll rotate through these over a period of semesters and let the students work on these reports. Okay, so Robert, you want to talk about the...
So, yeah, so since all faculty get access to this site free, you're set up for at least 1.5 years access. So if you actually use that code, when you go into, you have to create an account first. And once you go through the account, it'll ask you for a promotional code, and it is case sensitive. It's capital A, PS, capital R, lowercase o, capital C, lowercase s, capital S. And if you put that in there, you should get access to all aspects of the site for at least the next 1.5 years. If you runs out in 1.5 years, let me know and I'll refresh your membership on the site. Information, you can contact me at robert@justphysiology.com. Notice it says, if you look on there, it says full access for only $40. If you're in a class, it's actually $20 a student. We do things in terms of students buy it or institutions buy it. If institutions buy it, we can have discussions about a little bit of a discount with that. So I think that's it.
Also, Robert, the community colleges have a reduced rate.
Community colleges, yeah, I'm sorry. Community colleges have a reduced rate of $10 a student. We realize community colleges students are really probably stressed, and we figured let them try it out and, for that. So that's.
And I believe you're also persons in other developing countries. You have.
Yeah. Yeah, I know we have somebody from Azerbaijan. Please contact me and we'll work out a deal. I'd like to have a customer in Azerbaijan with that. So thank you, David, for reminding me about that. So I think that's it. I think that was our last slide. And if you want to stop sharing, and we can just sort of answer questions.
Okay, wonderful. Thank you very much. We have several in the Q&A. So I'll start with Joseph Rathners, who I believe is from Australia. And his first question is, I see temperature is in Fahrenheit. Can these parameters be changed to SI units?
Yes, we have, those are both available. I just happened to show it in that particular example. We used Fahrenheit, but we also can report it in Celsius. And
Yeah.
most of the parameters are available as milligrams per mil, millimolar, and other SI units.
Wonderful. And Rudolph has a question. PCO2, some textbooks claim a decrease only at high power exercise. Why?
I don't think I want to get into the research of that. One thing that we'll find is, and Robert can speak to this, this is a model, right? So this is one thing that's really important to understand about, and I try to impress upon my students. This is not like an animation, or it's not a preset list of values that will, when you put in, regardless of how you set this up, you're going to get the same responses out. It's 12,000 variables. We sometimes get responses that we don't expect. Sometimes, occasionally that's because the model isn't quite right, but frequently it's because our conceptions were too simplistic. And we find out that actually things are more sophisticated than we thought. Robert might want to speak for a moment about how Humod is used in, the engine for this is used in other applications.
Yeah, David, I appreciate that. One of the things you have to realize is a lot of this data we've gotten, I hate to say it, is from animal studies, because there's not a whole lot of human studies looking at giving them, fixing their insulin and giving them glucose loads and things like that. So what we have done, and we don't have it, it's not part of JustPhysiology, is we're actually creating virtual populations. So we can create 1,000 patients that are all slightly different from each other. And what we find out is that if you want to make them hypertensive, some of them will get hypertensive, some of them will not. Some of them will respond differently to different drugs. So the reality is this is an average, what we think is an average. We don't guarantee it, but we are using that actually two companies right now, we're actually using it for the start of clinical trials using virtual populations. Technology code is called digital twins now. Can you make a digital twin of a person, a physiological digital twin of a person? So that's one of the things we're working on right now.
Right. So just to clarify, so so that's the engine that underlies this. So it's used for research, used for clinical applications, used by government agencies to do models. When you use JustPhysiology, it doesn't have that randomization. So if you put in exactly the same parameters, you're going to get exactly the same result. We don't have that randomization, but you can do it slightly different times or slightly different treatment amounts, and you may get different results. So it's a good question as to whether it's accurate or not. And I tell my students it's not necessarily accurate, but the physiology, the interactions are based on research, the best we can do.
We don't want to put a bunch of virtual patients that are randomly assigned to people, because then nobody gets the same response, and that just would make David Julian's life a whole lot harder.
Wonderful, thank you. Joseph Rathner has another question. If we plan to use JustPhysiology as a simulated practical class activity, is it possible to get the actual measured values rather than simply the graphs, so we can ask the students to prepare grouped data?
Yeah, so as shown, you can export the CSV files. And so I have done things like that. You can put up a Google Sheet doc where the students could all input their data. And so you can get those as real numbers. But I should emphasize that one of the things about group data is that the issue is that if all the students do exactly the same inputs, then they're going to get the same result out. But if you give the students the opportunity to design their own experiment, then they'll get different results. Other things that we're not showing here are other things that I like to do are differential diagnoses, where we have some scenarios where there are 3 patients who might all have hypercortisolism, or they all have a cardiac valve disorder, and the students have to diagnose it, which patient has one type of pathology, which patient has another. And they have to design the treatment or the study in order to find the response. So anything that I can do that gets the students talking to each other, I like to do in my class.
Yeah, way I use it for our first year medical students. We give them a couple examples, an exercise lab and a hypovolemia, to sort of make sure they understand how to do that. And we actually have a series of patients that we break them into small groups that basically they have to diagnose the patient, determine the pathophysiology, determine what the treatment should be, and they have to give like a brief 15-minute presentation. So again, it gets the students engaged. And the realization is they want to be doctors, so this is what they're going to have to do.
Yeah, and just other things that we can do. Many pathologies can be simulated: heart disease, kidney disease. You can put the person on the top of the highest mountain. You can change the gravity. You can change the temperature. You can change the level of oxygen. Pretty much anything you can do. You can starve them. My students like to, if I give them a patient, and they like to just find out what it takes to kill the patient. That's one of the first things they do. Oh,
goodness. Thank you. All right, this was mentioned before, but I think it's worth mentioning again. How much is a subscription to JustPhysiology?
$10 for community colleges, $20 a student for undergraduate or graduate school, even medical school.
Thank you. And Lisa Anderson in Minnesota has a question. My understanding is the supplemental oxygen can increase hypercapnia, but it is not because of a loss of respiratory drive, but because of VA/Q mismatch.
Yeah, so the VQ mismatch question is interesting. The issue here is this question of giving supplemental oxygen to somebody who's chronically hypercapnic. That's the question.
Yeah.
So, and the idea is that people who are, this is not about just physiology, but the idea is that people who are chronically hypercapnic are using hypoxia drive for respiration rather than CO2. So the idea is if you give them supplemental oxygen, you're removing the hypoxia drive. And so what they used to teach clinicians was you never give somebody who's chronically hypercapnic supplemental oxygen because it reduces their respiratory drive. And it turns out that there was no experimental evidence for that. There's very limited experimental evidence for that. And that in fact, that the hypoxia, even when you give them supplemental oxygen, if the CO2 starts to rise, it's still enough to stimulate more breathing. So, and if they're really in severe condition, those of you that are clinicians out there will know you're going to have to intubate the patient anyway. So,
we really don't have enough capability to look at BQ mismatch in just physiology.
Great.
The lungs are not designed to be able to do that.
Okay, thank you. I think that answers her next question.
Yeah, that was the next question.
Yeah.
How long is a student subscription? 6 months, which we figured is a semester. If you decide you need it for a year, just let me know and we'll make some adjustments.
Okay. And is the code of, Naomi wanted to ask is the code available and what language is it written in?
So you can actually go to humod.org. You can download a version, which is a Windows program that runs on your computer. It is, you have to do a click through that states you're only going to use it for research and not for any commercial purposes. If you're looking for a collaboration about stuff, we can talk to you about that later on. But you can go through and play with it that way. I can tell you the interface is not very good. That's why we developed JustPhysiology.
yeah, and one of the key things that I wanted to do with that Humod is great. It's really wonderful and it's fast. The issue is that some of my students don't have Windows computers. A lot of my students use Macs, and so they have to, use the special Windows operating system. Some of my students at the time were using tablets. Also, the key thing is that with Humod, you can't plot one variable against another. Almost everything is against time. So with JustPhysiology, what I want to do, and we didn't show this really here, actually we probably could have, but we can do pressure volume loops, we can do, mean arterial pressure versus cardiac output, you can do blood glucose level versus insulin, and doing these kinds of plotting parameters against each other is a really great way of helping students see relationships between variables. And that's what we couldn't do in Humod, and that was one of the main reasons why we wrote the NSF grant to create JustPhysiology.
Wonderful, thank you. Joseph Rathner has another question. I'm interested in more information about how you set up custom experiments that you have described.
Yes, we do that. What ends up happening is that we go back and forth by emails, and I'll set something up so they can look at and to make sure it gets designed exactly, and they can run the simulations to see if it meets their needs. So yes, we do that.
Wonderful.
And one of the things we tend to do is we're going to work on being able to tag it for your institution. So that way, if your students go in, they say, oh, this is one that you developed. So therefore, they don't have to go down the full list of case studies.
And Xavier Navarro has a question. Is the software only available online, or can it be downloaded as an application for use in small labs?
That's what goes back to David's point. You can actually download it at humod.org. But I think
that's
Humod interfaces.
That's Humod only. So yeah. JustPhysiology can't be. No. It runs on servers, so it has to be run from the servers.
Yeah.
Great, thank you. Terry Sweeney asked if one wished to use this for a primarily lecture course, such as CV Physiology. Have you considered a short-term subscription that might allow it to be incorporated in, for example, the last 2 or 3 weeks of the course?
Dr. Sweeney, if you will drop me an email, we can have a discussion about that. I'm open to sort of helping people get access to the site, and we can talk about that.
Wonderful, thank you. And Ed Merritt is asking, can the raw data from each model be exported from the different simulations?
Right, so I think he's referring to the output. All of the output variables can be exported.
Wonderful.
All of the displayed variables can be exported.
Great.
Not all 12,000.
Right.
Great. And Jason is asking, is this strictly an online platform, or is there the capability to hook it up to equipment for real-time experiments?
It is only an online platform. We don't know what equipment you want to have, but no, it's just strictly an online platform.
But I'm sure Robert would be, HC Simulation, I'm sure would be willing to consider anything at this point. Always open to what's in the future. We have had conversations with people who wanted to this up to simulators, for example, right? So
Yeah, we actually, that's the other thing. Just to give you an example, we actually have DoD projects for mannequins for in virtual reality that we're in the process of developing. We actually have some developed, and we're actually going into VR for training for medics in the battlefield.
Great, fantastic. And then someone said, for a model with 12,000 variables, has validation against animal data presented difficulties? Has there been clinical validation?
Let me say this, against animal difficulties, I will say this, as a physiologist, there's always challenges comparing one experiment against another one, because you don't necessarily, it may be initial conditions or how the procedure was done. We have a set of simulations that we run to make sure things are looking reasonable with that. Clinical data, my colleague John Clemmer here is in the middle of finishing a study in which he basically looks at, compares Humod to African-American males, African-American population, I'm sure it's males, 165 males that were had hypertension and stimulated and got 3 years, had calcium channel blockers for 3 years. He's actually done that simulation. He can actually parameterize the model, and he gets very similar responses to what they get in the real patients.
So, and actually to emphasize, so in Humod, you know, every variable, every response is based on a publication, right? So this is over the last 30 or 40 years. All the parameters are based on, whenever possible, some kind of research. Getting them to work together, I don't know how Robert and his team actually does that. That's above my pay grade. Most of it works pretty well. It doesn't always, right? Sometimes you get really crazy responses when you try to do something that's just unrealistic. And my students don't mind that. I try to emphasize this is not a real person, it's a model. And the old trope is that all models are wrong, but some are useful, right? So this is useful for teaching. And in fact, sometimes when it deviates from expectations, that's useful to discuss as well. But there are a number of publications. I mean, so the Humod side, there are many publications in peer-reviewed journals using Humod to make predictions about patient outcomes. So JustPhysiology not, but the engine underneath it, yes.
Wonderful, thank you. And Erin, I think it's just worth repeating again, is the subscription price per month or per year? You mentioned a 6-month subscription. Can you get a year?
Yeah, we can have, contact me, we'll talk to you about a rate for a year.
Wonderful, thank you. And here's a good question from Reem in Egypt. Does the software provide an explanation for the responses or a link to the reference?
I will say no. We actually have about 3,000 papers in the files that we use to do this, so it's a little hard to give those references with that where we did the model. So no, we do not have that.
That might be a additional assignment for the students to go find a reference. So another question, in medical school, every in-person contact minute is precious. We are constantly being asked to cut. What did you cut to be able to include these simulation labs?
So I teach in physiology, the first-year medical physiology course. We have always had some time for small group discussions. So basically we do 3 for the class that participate in 3. One of them is the hypovolemic treatment to learn how to use the model and so forth. And then we have 2 patients. One's a cardiovascular patient. We actually have 4 different cardiovascular patients, and they're all broken up into small groups. And we do that 4 times a week. So each is about an hour-and-a-half session that is spread out. And we do an interview again about hour-and-a-half, 8, excuse me, it's actually 8 different sessions. We have 2 instructors, and we do that for about 3 to 3.5 hours twice a week.
Wonderful, thank you. And Brian Dewar wanted to know, with pressure volume loops, can you then modify MAP after load and see how loops changes? Yeah,
so and one thing this is actually useful to talk about. So there are actually multiple models. So the general Humod model that underlies this doesn't model individual breaths or cardiac contractions, but we have isolated models. We have an isolated model for a neuron, and we have an isolated model for the heart, and you can do that. So I'll just real quickly, I'll just pull up a different one just to show. So this is an example of an isolated heart model. And actually, let me go. So we can change, we can see the ventricular pressure, ventricular volume, aortic flow, and we can generate a pressure volume loop here. There are other parameters that we can change, preload and afterload, and you can change the conductivity of the valves. So you can induce mitral regurgitation or aortic stenosis, aortic regurgitation, mitral stenosis, and you can see the resulting changes in the pressure volume loop. So one activity I do is I give my students 3 patients, all of whom have valve disorders, and I give them a normal patient. And I ask them to go through and experiment with this and to then diagnose. They can see, they can play around with the valve conductances, and then they try to diagnose the valve disorders. This I don't have built in the preload and afterload, but that is possible in the cardiac cycle model.
Wonderful, thank you. And actually, this was asked a while ago in the chat. So going back to the chat, Clayton Peter wanted to know, why is the x-axis over 10 hours, even though it is a 2-hour simulation? And then why does the plasma glucose respond at 6 hours?
That was, yeah, so that was, what was happening was the x-axis is just like on a chart recorder. It's scrolling out as you run the simulation. So we started off doing a 6-hour fast. Then we did the oral glucose tolerance test, and then we followed it. And so each time we set it, there's a time setting. Students or you can, depending on what the available times are, you can run the simulation for a second, a minute, 10 minutes, a week, 4 weeks, and the x-axis adjusts. So I think that was just we didn't explain that clearly what was going on the x-axis there. Just think of it like a chart recorder.
Wonderful, thank you. And then Terry Sweeney mentioned he's done ski patrol, and the internal bleeding scenario would be of great interest to this type of first responder training.
Yeah, I've used this. I mean, there are a variety of people. I mean, I've actually used Humod with middle school students, where all I did was this is when they were all trapped during COVID, and I got contacted by somebody in Florida. And so I just had the students jump up and down and notice that they were breathing faster. And then we just pulled up a really, really simple model where we could just show the more they exercised, they could look at their heart rate and they could look at the breathing. And so there's a whole range of students. And then you can run it, obviously, with all of the cardiovascular medications that you can give. You can do that with the most sophisticated physiology graduate students as well. Certainly challenges me a lot, and I have a lot to learn.
That's wonderful, thank you. And then someone asked, to accommodate for expanded complexity, is development of the underlying engine ongoing? Is this done mostly at HC simulation?
It is continuing ongoing. One of the things we've developed, we haven't put in just physiology, is we actually have a model of heart failure with preserved ejection fraction, which is a big topic nowadays. So we're continuing doing that, adding content to the model. Basically the needs for our customers. What our customers say, do we need to do that? We take that same version, ended up putting this in just physiology. We do have one model that we will not put in just physiology. It actually has 40,000 variables and parameters. Basically, it's more anatomical, but it runs slow, and we don't want to do anything that takes a long period of time to run.
Great, thank you. And the Rudolph asked, PV loops, what about heart failure? I think you just said a little bit about that.
Well, that's in the whole Humod. I don't think that the isolated version.
Yeah, I don't think you can model reduced contractility in the PV loops. You can in Humod, and so you can induce heart failure through a number of different mechanisms, pathologies in Humod, and you can watch that over time. Not with the PV loops, I think. I don't think there's a contractility.
Yeah, I have to go back and look at the Humod and look at that. We might be able to add that if we do when we add the pressure heart failure with preserved ejection fraction into that.
Wonderful, thank you. Ahmed asks, it appears most of the simulations are patient/clinically based. What about simulations looking at basic physiology content such as memory potential?
Right. So so we do have an isolated nerve model, and I'll share that again. So we emphasized a lot of the more sophisticated ones here, but we have a lot of basic ones that we're adding to those still, actually one of my summer projects. So here are some basic models. We do have neuron action potential. So we have the Hodgkin-Huxley model, and you can stimulate nerve action potential, can look at changes in current, et cetera. And we have other more sophisticated ones where you can look at the number of active versus inactive sodium channels to try to get a sense of when the relative and absolute refractory periods transition, or when it transitions from absolute to relative. We have other ones on, I have to refresh this. We have some more ones on blood flow and blood pressure, cardiovascular reflexes, gas exchange. These are just simple ones like mechanics of breathing. Here, where in this one, we just have the person do exercise and they can look at the changes. Actually, that's not the one I wanted to show. I wanted to do ventilation alveolar gases. In this case, the person controls, the student controls the tidal volume and ventilation rate, and then they look at the effects on alveolar PO2 and arterial PO2. So trying to get at some of the basic ideas that are covered in typical textbooks for lung function. And then we have others where you can change diffusion parameters, you can change the PO2 levels. So we do try to cover, in addition to having all these fancy things that we can do, we do try to have some basic models where students can play around with the parameters that they read about, like heart rate, peripheral vascular resistance, stroke volume, ventilation, tidal volume, and then see what the effects are on cardiac output or alveolar blood gases or arterial blood gases.
Very cool. Thank you. Monty Krieger wanted to know, can you set up a set of parameters in advance, like groups of the same question, and then just plug them in quickly in class? That sounds like a lab class.
Yeah, so I'm not quite sure I understand, but I mean, we do certainly have these where we have case studies and they're set up as we show with a series of labs that address a particular question. And each time you cycle to a different lab, then it can have any number of preset conditions, so default conditions, or we'll give the students instructions as to what to set it to, and we can customize those. I'm not sure if it addressed the question.
Yeah, I'm not sure either, because in our idea, parameters are the M and B on the y equal mx plus b's equation for a curve. So that actually shifts the curves, any relationships we have, to the left or right or up or down. So we really don't do that. That goes back to our whole idea about virtual patients, making everybody a lot of different versions of the model within the same thing. And we really don't have.
I interpreted parameters to mean more the inputs. So like tidal volume, heart rate, blood loss, body temperature, ambient temperature, altitude, things like that.
Yeah, if we do it like that, then we can do some work with you, Monty. If you have something you want, let you develop it, and development's fairly quick.
But otherwise we just say, you know, I tell the students what to set as their starting conditions and how long to run the simulation.
Great. Thank you. And then someone else asked, are there 1D, 2D, or 3D spatial models in the underlying engine, or are the models lumped parameter models?
The model is a lumped parameter. We don't do any spatial models in this particular version.
Great. Well, I think we've answered all of them.
Again, if you have any questions, don't hesitate to drop me an email. As I said, it's robert@justphysiology.com. David will be glad to answer things. I'm putting him on the spot. djulian@ufl.edu, or contact me and we can get you in contact with David to sort of see how he uses it. He's been using it, what'd you say now? 7 years.
Since we first started making it. I mean, I was using QCP and Humod before then, so.
Yeah.
Nice.
Yeah.
A lot-
And
a lot of-
I'm very interested in hearing ways that people use it and other ideas people have for using it. I'm always trying to find different ways to try to engage the students with it. I'm gradually getting better. The students are liking it more and more. Yeah, so anybody who has ideas or any way you use it, be happy to learn, and maybe we can build that into models we make available for other people too.
So please provide feedback.
Absolutely.
Yes.
Well, I wanted to say thank you very much to Dr. Hester and Dr. Julian for providing us with your expertise and insight into novel physiology education technology. I also want to thank HC Simulation for their support of APS awards and activities, as well as all of you in the audience who've joined us today, especially all those wonderful questions. And before you sign off, we hope that you will take a few minutes to complete a short survey about this session and your interest in future webinars.
And we'd like to thank Patricia for being an excellent
Yes.
moderator.
Thank you,
My
Patricia.
pleasure. My pleasure.